Hello guys, welcome to my YouTube channel at John Pharmacy. Biochemistry, that is Biochemistry and Clinical Pathology of D Pharmacy second year. Those who are a new syllabus, second year students, for them a very important video, very important video. In this video, you are going to get the notes of the first part. This one is the part 1. Okay, very good notes by practicing only these notes. Two notes I am preparing for Biochemistry and Pathology. One is part 1 and part 2. Today we will discuss some of the important questions in part 1 with answers, and in part 2 we will discuss some other important. If you practice that, means you can easily pass your biochemistry exam. Challenge it is. Yeah. Let's start with the first question. At the scope of biochemistry in pharmacy. See, scope of if you want the notes, means you can WhatsApp me. The cost of the notes is very less. Yeah. It cost only 51 rupees. You can get it only 51 rupees. What is the scope of biochemistry? See, importance of biochemistry. Biochemistry is one of the branches of chemistry which deals with the chemical basis of life in plants and animals. Yes, it is a branch of chemistry. It deals with the chemical basis of life where in plants and animals. It is used in clinical diagnosis, then manufacture of various biological products, treatment of disease in nutritional agriculture. It tries to explain life in terms of a biochemical reaction, including other sciences as anatomy, medicine, agriculture, pharmacy. And it is also used as a nutrition deficiency. Means in the present scenario, many people rely on taking multivitamin and multimineral, okay, for better health. The function and role of the vitamin in the body are described only by the biochemistry only. Today, everyone is taking multivitamin tablet for the weakness. These multivitamin, multiminerals, these are derived from the biochemistry only. Hormonal deficiency, there are many disorders due to hormonal imbalance, and in especially women and children, the formation role of hormones in the natural body function is taught in biochemistry, by which the physician can understand the concerned problem during treatment. This is also on the basis of the biochemistry. This is the scope. This you write down. Clear? This is the scope of biochemistry. Next one, define and classify carbohydrates. See, they are carbohydrates as they comprise carbon, hydrogen and oxygen. They contains carbon, oxygen, hydrogen at their chemical level. And carbohydrates are essential nutrients that includes sugars, fibers and starches. They are found in grains, vegetables, fruits, in milk and in other dairy products. They are the basic food groups which play an important role in healthy life. Yes, they are found in many sugar products, in fibers, in starches, in carbohydrates, okay, like in rice, potato. There are all good amount of starch is present. Now this classification, carbohydrates is classified as a monosaccharide. Example is glucose, disaccharide, sucrose, oligosaccharide. Example is oligofructose. Polysaccharide, example is cellulose or starch. These are the four important classifications of the carbohydrates. Now structure of glucose and fructose. See, this one is the structure of glucose, this one is the structure of fructose. Okay, remember this one, fructose and glucose. Somewhat they will ask from here only. Okay, just they will means they make some twistness, you think. If the question paper is difficult, means they make intertwine the question, but definitely they will ask from this only. No out from this. Remember this point. Next, biological role of carbohydrates. Biological role, significance, importance, everything is same. Whatever they ask in carbohydrate, you write this one. Okay, if you don't know anything, means you write this one. Importance of carbohydrates. See, you know that provides major source of energy, carbohydrates. Then also store energy as a glycogen in liver and muscles. Then protein sparing action. Then reduce breakdown of fatty acid and preventing ketosis. It will reduce the breakdown of fatty acids. And also involved in the biological recognition process of the immunoglobulin. Okay. Then what are defined and classified proteins? See, proteins are extremely complicated and nitrogenous molecule made up of variable number of amino acid. Okay, residue joined to each other by a specific covalent bond called a peptide bond. Proteins, they are the group of amino acids which are joined by the peptide bond. 20 amino acids which have been found to occur in all protein. That's known as standard amino acid. Classification is based on the two types mainly. Here one is globular and one is a fibrous protein. And test and significance of protein. One is a color reaction test is there for protein. See, amino acid, if you add reagent, means protein color complex will be given. Complex can be used for both qualitative and quantitative detection of the protein and identification of the amino acid. If amino acid is present, means it is a protein. Remember, reactions are based on the amino acid residue and peptide bonds. The Millians-Nesse reaction. Here we are using Millians-Nesse reagent, 1% sodium nitrate. Add 1 ml of Millians reagent to 5 ml of test solution. You take 5 ml of test solution, you add to that 1 ml of Millians-Nesse reagent. Place the tube in boiling water for 10 minutes and cool the contents in water bath for 5 to 10 minutes. After that, add 1% of the sodium nitrate. A deep red color indicates a tyrosine or other 3,5 substituted phenol, but tyrosine is the main product when we are doing this one Millians-Nesse test. Tyrosine, you know that tyrosine, it is the protein, amino acid. Are you getting? So this test detects the presence of protein. Next, functions of protein. A protein is vital in maintenance of the body tissue, including development and repair. Protein is the major source of energy. Protein is involved in the creation of some hormones, help control body functions, and that involve the interaction of the several organs and help regulate the cell growth. Yes, protein produces enzymes that increases the rate of chemical reactions in the body. And proteins transport small molecules to the organism. Then hemoglobin is the protein that transport oxygen to the cells, and it is called the transport protein. Hemoglobin, it is also a protein, it transport the oxygen. Proteins called antibodies help rid the body of the foreign particles and help prevent the infections, illness and disease. The last point is protein help to store the other substances in the organism. For example, iron is stored in the liver in the complex with the protein ferritin. Okay, it helps to store, transport, fight with the infection. Okay, it provides a good nutrition and malnutrition of protein. Seventh question is a malnutrition of proteins. See, malnutrition as a cellular imbalance between the supply of nutrients and energy and body's demand for them to ensure growth, maintenance and specific functions. If body is unable to grow or it doesn't showing the function, means you think that there is any one deficiency. Means the material which we are taking inside the body, that is in very less amount. Anything it is, protein, mineral, vitamins, anything. If that occurs, means malnutrition. Now here we are discussing about the protein malnutrition, disorders of the protein malnutrition, protein energy malnutrition. Okay, first one is a kwashiorkor or marasmus. The next one, undernutrition of vitamins and minerals, obesity. These are the three important malnutrition problems. Kwashiorkor, see, it is the form of a severe protein malnutrition characterized by the edema, means swelling, and an enlarged liver with fatty infiltrates. In fatty acid, what happens? There is a deposition of the waste products. In means cholesterol deposition. There is a, because of insufficient proteins, what happens? There is a deposition of fatty infiltrates. It is caused by the sufficient calorie intake. There is increase in the calorie intake, but with insufficient protein consumption. This is very important. And kwashiorkor is a serious condition that can happen when a person does not consume enough protein. Okay, severe protein deficiency can lead to fluid retention. You observe that fluid retention means edema, swelling. Swelling may be where? Mainly swelling on the stomach region, liver side, which make the stomach look bloated. How the pregnant woman's stomach will be look bloated, no? In the same way, this kwashiorkor patients it looks like that. Very, very, very popular in Africa because of malnutrition. Next one is a marasmus. The term marasmus is derived from the Greek word marasomos, which means the wasting. Here marasmus is the form of a protein energy malnutrition characterized by the energy deficiency and abnormal thin or weak, means emaciation, thin, person is thin and weak, very weak, primarily caused by the energy deficiency. Marasmus is characterized by the stunned growth and wasting of muscle and tissues. Here what happens? The muscles get wasted because of the energy, insufficient energy supplied at all. And marasmus especially usually develops between the age of 6 months and 1 year in children who have been weaned from breast milk or who are suffering from the weakening conditions like chronic diarrhea. For them, this one is very attractive. Then define and classify lipids. See, lipids may be regarded as organic substances. Relatively, they are insoluble in water and soluble in organic solvents. Actually, are potentially related to fatty acid and utilized by the living cell. These are the insoluble in water but soluble in organic solvents. The functions of this lipids. Lipid is stored in adipose tissue where it is also serve as a thermal insulator in the subcutaneous tissue. Main function is it provides heatness. Fatty acid derivatives serve as a vitamin A, vitamin D, E and K or hormones. It acts as an energy or food reservoirs, like it will reserve the triglyceride level. Several proteins are covalently modified by the fatty acids. Structural components and biological membranes, they are made up from the cholesterol, that is lipids. Okay. Classification, broadly they are classified into 5 types. One is a simple lipids, complex, derived, neutral, miscellaneous. What are these simple lipids? Simple lipids, see, esters of fatty acids and glycerol, mainly fats and oils and waxes. Fats and oils, these are ester of fatty acids and glycerol. The difference between fats and oils is physically there is a difference. Okay, you know that oil, it is somewhat solid in nature. Fat is somewhat solid in nature, yellowish in nature. But oils, they looks like a light yellow, but they are liquid in nature. This one is different. Waxes, esters of fatty acid plus alcohol other than the glycerol. Glycerol is absent in waxes, but alcohol is present. Cetyl alcohol is the most commonly used. Cetyl, you are using the moisturizer, no? Yes, this one. These are the simple lipids. Now complex or compound lipids, esters of fatty acids, alcohol, other group like phosphate group, nitrogenous group or carbohydrate, protein, any other group. Beyond this, fatty acid and alcohol. Based on the group present, they are further classified as a phospholipid. Here it is fatty acid, alcohol, phosphoric acid as a nitrogenous base. Based on the type of alcohol present, glycerophospholipids contain glycerol as an alcohol, and phenogophospholipid contain phenogosine as alcohol. Example, phenogomyelin. You write this one, you definitely will get the marks. Then glycolipids, fatty acid plus alcohol. Okay, it is the third type of the classification. Glycolipids, fatty acid plus alcohol plus carbohydrates as a nitrogenous base. Glycoprotein, glucose means carbohydrates. They contain spinogosine as alcohol, and here is known as the glycosphingolipids. Then lipoproteins, our last classification is lipoproteins, macromolecular complexes of the lipids with proteins. That LDL means low density lipoprotein, VLDL very low density, then chylomicrons, HDL high density lipids. Next, disorders relating to this lipids are, that is hyperlipoproteinemia, then the hypolipoproteinemia, fatty liver, obesity, cachexia, anorexia nervosa, xanthomatosis, atherosclerosis. These all are the diseases related to the lipid metabolism, lipid problems or lipid disorders. Define purine and pyrimidine bases. Purine and pyrimidine, simple. These purine and pyrimidines are nitrogen containing heterocyclic compounds whose rings contain carbon and nitrogen. They contain carbon and nitrogen. The planar characteristics of purine and pyrimidine facilitate their close association or stacking with stabilized double standard DNA. Mainly this double standard DNA stabilized, prepared by this purine and pyrimidine. They are also referred to as the nitrogenous bases. Nitrogenous bases are the unit point for this DNA. They are the main for this DNA preparation and major components of the nucleotides that built DNA and RNA. Okay, that is known as this nuclear bases. Purine and pyrimidines are the nuclear bases, nitrogenous bases. Biological function of nucleotides. See, building blocks of nucleic acid, involved in energy storage, that is muscle contraction, active transport, maintenance of ion gradients. Then they help for the activated intermediate in biosynthesis like glucose synthesis, adenosylmethionine, compounds of coenzyme NAD, NADP, FAD, FMN and coenzyme A, metabolic regulators like CAMP, GAMP, phosphate donors in a signal transduction ATP, and regulation of some enzymes. This nucleotides are very important. Next one is define components of nucleosides and nucleotide. Components, composition, nucleosides, what you are observing now, it is mainly result from the linkage of one sugar moiety with a purine or the pyrimidine base, okay, through an N-glycosidic linkage. N-glycosidic linkage. This is the sugar moiety with a purine or the pyrimidine. With a which one linkage? N. This one is N-glycosidic linkage. This is a nucleoside. Purines bond to the C1 carbon of the carbon at their N9 means nitrogen 9 atoms. Pyrimidines bond to the carbon 1 of the sugar at their N1. In purines, carbon means this sugar molecule joined at N9 atom in nitrogen 9th atom, but in pyrimidine carbon 1 joins at the N1 nitrogen 1 atom. Next one is phosphate groups. These phosphates can be bonded to either carbon 3 or the carbon 5 atoms of this nucleoside, either in purine or pyrimidine. Nucleotides, it is the ribonucleic acid is a polymer of the nucleotides RNA. DNA is a polymer of the deoxyribose nucleotides. Both are very important. Both deoxy and ribonucleic acids contain this, both it should contain adenine, guanine and cytosine. Yes, ribonucleic acid contain uracil, deoxyribose nucleic acid contain thiamine. This is one major difference here. Next, this is all about the nucleotide. Draw structure of DNA and Watson-Crick or Watson-Crick model. Both are same. So DNA is a double helical standard molecule that is twisted into a helix. DNA are macromolecular structures, polymers formed from the nucleotides. DNA, you know that I have told you, DNA, deoxyribose nucleic acid and ribose nucleic acid. It is very important. Preparation is mainly by the nucleotides only. DNA is a complex molecule that contains the information necessary to build the organism. This one is the structure of the DNA. Draw this structure. This structure is enough. Only this structure you draw. Very important. Types of DNA. DNA types: A DNA, B DNA and Z DNA. These are the three important types of DNA. Functions of DNA. DNA replication. It provides the information inherited by daughter cells or offspring. Okay, yes, daughter cell to the offspring. It will provide the information like replication. And storehouse of genetic code. The DNA holds the information for all the proteins to be created for the cell. The bases are grouped in triplets called the codons that code for the specific amino acid. The whole genetic code stores the information for all cell type to reproduce. Mainly they store the genetic code information, which is very important for the reproduction and all. Functions of DNA in mutation and recombination. Mutation means sudden change. During DNA replication, different DNA segments can be spliced through gene linkage. The process can create one combinations of character in offspring. Character or traits, both are same. If the protein helps the species survive, it may evolve over time. However, some mutations may be non-lethal, means non-beneficial. They cause the death of the many cells. This which one? Some of the mutation. But somehow mutations will help for the development. Remember this one, mutation and recombination. What is the function in the DNA? Function of DNA in gene expression. Each cell contains a full set of genes. Cells from different tissues and organs function differently. Function of the hand, function of the leg, they all are different because the tissues are different there. They behave the different functions. Okay, the reason is that only some of the DNA of each cell is used to make the proteins. All cells won't produce the protein. Only some of the DNA, all DNA won't produce the protein. Some DNA will produce the protein. Difference between the DNA and RNA. DNA and RNA. See, sugar moiety is a deoxyribose here. Here sugar moiety is ribose. In the name itself, deoxy or ribose. Uracil here is absent. Here thiamine is present. Uracil is here absent. Here Thymine is absent. Where? Sorry, in RNA thymine is absent. In spite of RNA, what is present? Uracil is present. In DNA, in spite of this uracil, what is present? Thymine is present. You can write it down, inverse and write. You can write in DNA thymine is present, in RNA uracil is present. Then double standard molecule DNA. RNA is a single standard molecule. Sum of the purine bases is equal to sum of the pyrimidine bases, that is Ag is equal to C plus T. Sum of the purine bases is not equal to sum of the pyrimidine bases. Why? Because means A plus G, yes, equal, but it's not equal to C plus T. Remember this one, because here thymine is absent, uracil is present, so C plus U, that is a cytosine plus uracil in RNA. Resistance to hydrolysis by alkali because of the absence of hydroxy group on 2 carbon atom of deoxyribose. Here hydrolysis, resistant to the hydrolysis. Here because of presence of hydroxy group, carbon atom on ribose easily hydrolyzed by alkali. This ribose can be easily hydrolyzed, but it won't be easily hydrolyzed. They won't break down, they won't hydrolyze. Why? Because in DNA what happens? There is absence of the hydroxy group. But in RNA, presence of hydroxy group. Yes, these are the very important 12 questions in biochemistry. You practice these questions. Part 1, inshallah, soon we will discuss about the part 2. Thank you, and subscribe our channel and try to share this video with your friends. Thank you.